15th August.
Nisha, ma'am joined. Shall we start?
Nisha, am I audible? Now okay?
Shall we start, ma'am?
Hold on for a while, ma'am. We'll start recording, then you can start.
Question does start recording.
Yes, ma'am. Yes, ma'am. Give me a moment.
Madhusudan sir is ready, ma'am?
Ready.
Yes, ma'am. Thank you.
Yes, madam.
Ma'am joined.
Hello?
Hello.
Shall we start?
Okay. So I just wanted to check whether I'm audible. Okay.
Yeah, ma'am. Yes. Dr. Priya, ma'am, shall we start, ma'am?
Start, Nisha.
Good evening, everyone. The day starts with one beautiful quote. The capacity to learn is a gift. The ability to learn is a skill. Willingness to learn is a choice. I hope everyone is very willingly enjoy our Atal FTP. Once again, I welcome to the Atal FTP IOT driven wearable devices for healthcare and defense application. Today's session is about advance in bioanalytical techniques with Enzymes Biosensor. Today, now I call upon Mr. Madhusudan sir to introduce our resource person. Please, sir.
Yeah, thank you, madam. Good evening, all. This is Dr. Madhusudan Kulkarni. So I am very happy to introduce today's chief guest, Dr. Kanchanmala Deshpande, Assistant Professor, School of Chemical Science, Goa University, Goa. So her qualification is PhD from BITS Pilani and Masters and Bachelors in Chemistry from Nagpur University, Nagpur, India. And she is currently working as Assistant Professor, Biochemistry Department in School of Chemical Science, Goa University, Goa. And she worked as Research Associate and Senior Research Fellow in Department of Chemistry, BITS Pilani, Goa campus, and Technical Assistant in Department of Metallurgy and Material Science, IIT Bombay. Project Assistant and Research Associate in Department of Chemical Engineering, IIT Bombay, and a Junior Project Fellow in National Environmental Engineering Research Institute, that is called NEERI, Nagpur. And her contribution is 9 international publications, 1 book chapter, and 15 conference work presentation she presented. And including these other academic achievements also she got, and she got her project research grant by DST in women's in research, and she got postgraduate diploma in ecology and environment from Indian Institute of Ecology and Environment, New Delhi. with distinction, and Bachelors of Education from Nagpur University, first class. And I welcome Madam. I welcome Dr. Kanchanmala Deshpande once again. Thank you all, and thank you for giving me this opportunity. Thank you, Madam.
Hello.
Hello,
Uh,
Nisha.
Yes, ma'am.
Start the session.
Okay.
Yeah.
Okay. So,
Hand over the session to ma'am.
okay.
Ma'am, we once again welcome you, ma'am. Please start the session, ma'am.
Okay. Thanks a lot. Thanks a lot to all of you, especially Priya, Nisha, and Dr. Madhusudan. Really, I'm really thankful to give me this opportunity. And I'll share my screen and then I'll start with my presentation. I hope you all are able to see my screen. Hello?
Yes, ma'am.
Yes, ma'am. Yes, ma'am.
Okay. So, okay. So, Dr. Madhusudan has already given a brief idea about me. I'm currently working in the School of Chemical Sciences in the Goa University, and I'm basically an analytical chemist, like working in the field of biosensors. So my today's sessions, which I'll be taking care, I'll be discussing more about the all these bioanalytical or analytical techniques which are used, especially with your theme of your FDP, which is regarding the healthcare analysis, samples from healthcare sector, and as well as for the this military application or defense application. And then that's all what I am planning to discuss today. Okay. So like need of analytical or bioanalytical tech tools has been emerged, like has been increased tremendously over last few years. Now the reason behind increasing the significance of these analytical tools or bioanalytical tool can be, there are a number of other reasons, but the significant reason, if you figure it out behind this, is that these because we are getting exposed to large number of chemicals on a day-to-day basis, and which exposure to these chemicals leads to the different diseases which are quite which are occurring quite commonly in our surrounding. Now this particular picture I just pick up to show you that even if like we are purchasing something from the market, basically from each of those products we are getting exposed to n number of different chemical. Those chemicals, basically I would like to say, particularly for those chemical, like we can say that they are unknown chemicals. The chemical name might be known, but the toxic impact of all those chemical is not known. We don't know that over the period of time what impact these chemicals are going to cause us. So because of exposure of these large number of chemicals and their unknown effect or ill effect on our bodies, a large number of people are suffering from various metabolic diseases, and that's why the need of the sensors in healthcare sector has increased tremendously. Now my today's presentation, I'll be discussing with these few guidelines. First part of presentation, initially I'll be briefing you with analytical and bioanalytical techniques. I know in this particular FDP we have people from different backgrounds, so just to give you idea about the analytical technique, I have included these few topics. Then I'll be discussing about device and instrument which are used for the healthcare field. Then I'll move to the sensor. After briefing a little bit about the sensor and chemical sensor, I'll discuss about the chemical sensor with some of my published work regarding chemical sensor. And then I'll be giving you a brief idea about biosensors. With that, I will conclude this first session. Now, before we start discussing about sensors or biosensors and what are their application in the healthcare sector, we need to understand the basic idea behind the Analyte monitoring devices. Now, these are some of the very common terms which we use in our analytical chemistry or biochemistry. Many of you might be quite familiar with this, but to give a clear idea, I thought of including this very preliminary slide. So, during the entire discussion, when I'll be discussing about, like saying Analyte, that Analyte will be something which is interest of our analysis. Like here, I have shown you the like example where the normal blood glucose and high blood glucose is shown in the human body or arteries or veins. So when we are talking about the glucose in the blood, and if we are interested in measuring the glucose in the blood, that glucose will be the Analyte. And now that glucose is present in the blood, so anything other than the glucose which is present in that blood, like maybe WBC or plasma, everything apart from the glucose, it will be a matrix. Now, why I have included this slide? Because we are going to discuss about the sensors which will be able to monitor these Analyte, like say maybe glucose, but the monitoring or the analysis of these parameters are highly dependent upon in which matrix they are present. Like the constituents of blood are definitely going to create problem or may interfere in the analysis of glucose. That's why these terms such as Analyte and matrix are highly important when we are thinking of developing some Analyte monitoring devices. Now, as I told you, as Dr. Madhusudan has given you idea, I am basically an analytical chemist and working in this field. So I'll just try to give you idea that how the analytical chemistry can contribute in this field. So basically, analytical chemistry is a scientific discipline, and this discipline deals with developing the different instrument and figuring out the composition and the nature of matter. Now, when we are discussing, or when you consider the entire chemistry as a whole, it discusses about the various components. You can see here, you have synthesis as one of the major component, which is normally handled by...
Ma'am,
Yeah?
sorry for the interruption.
Yeah.
Show your PPT, madam.
My PPT is not visible?
Oh, no, madam. We can't see your slides.
Yeah, ma'am. We are seeing your screen. Your screen is sharing, but PPT is not opened, ma'am.
So what is sharing there then?
Then
the
you are
PPT,
just library only opened, ma'am.
Okay. Just...
You just selected that.
Here I could see that it was sharing. Anyway, I'll just try once again. No? Just give me a minute.
Okay.
Yeah, ma'am. Now it's...
Now it is visible.
Okay. So I'll move to the first slide once again, and I'll quickly go through the things. Okay. Sorry that... anyway, so this was the figure which I was discussing initially, that when we are on a routine basis, how we are getting exposed to large number of chemical, and that's why we need to have those kind of analytical devices or tools which will monitor these toxic compounds which are present in our day-to-day routine, on which we are getting exposed. Now, I would like to emphasize here that all these chemicals which we have written here, their concentration might be very less, but because their ill effects are not known, that's why some sensors need to be developed for all these things. And that's why the significance of developing sensors for health sector area has been increased tremendously over the last few decades. This slide discuss about how I'm going to discuss go with this particular first session of your presentation. And then I would like to start. I gave you brief idea about two important terms which are routinely used when we discuss about developing some sensors for a particular component or biochemical component. So like analyte is something which we wanted to measure. Like in this particular example, in this figure, you can see it's a sugar, and the matrix will be everything other than the sugar. So the effect of matrix in analyzing the sugar will be tremendous, and that's what we need to figure it out when we are discussing or when we are planning to develop the sensor. Now, chemistry plays a significant role in developing these kind of sensor. Like you all might be quite aware about the organic chemistry. We discuss about the synthesis of large number of compound. Then inorganic and physical chemistry, which might be discussing about the theory. But one more component of chemistry is highly important, which discuss about the tools which we can utilize to identify the compound or develop the techniques by which we can figure it out that what is present in the sample and how much is present in the sample. So that particular tool which gives you idea about the quantitative, qualitative, and structural determination of the sample is known as an analytical chemistry. Now I'll give you brief idea. Whenever we wanted to figure it out that what is the composition of the sample, that will be a qualitative analysis. You get idea about what is the quality of that sample. Then when you wanted to figure it out that at what concentration that particular toxic compound is present, then that is a quantitative analysis. And there are some tools, like if I'm analyzing a completely unknown sample about which I don't have any other idea, in those cases, I need a tool which will give me idea about the structural identity of that compound, and that those are the tools like which will help me to figure out structural determination. So these are the three significant ways by which analytical chemistry is useful for us. Now let me tell you, whenever we are routinely developing a sensor and all those things, we will be discussing more about the qualitative or quantitative nature of this tool. So like if you are developing a sensor, that sensor will be able to figure it out, will be a good qualitative tool when it is able to give us idea that whether that particular compound is present or not, yes or no sensor. And if that particular sensor is helpful to identify how much it is present, then it is a quantitative sensor. Now, to develop a sensor as a quantitative or a qualitative sensor, you need to have some standards. What are the standards? Standards are some reference compound, like pure compounds, with which I am going to compare a sample. So always remember that whenever you are analyzing a sample, you need to have a reference standard for that particular sample. Unless and otherwise, it is not possible for you to analyze the actual sample. Now, with this brief introduction about role of chemistry or analytical techniques in developing sensor, let's proceed further. Now, what exactly does this analytical techniques, or to be precise, bioanalytical techniques help us in developing sensors? Because they help us to give us large amount of biochemical information. We are discussing preliminary about the healthcare sectors and all those areas where the samples will be from some biological origin. So these sensor will be helpful to get some biochemical information, that too with the minimal sample preparation, minimum time and human resources will be utilized. And that's the purpose of a very good tool or technique or a sensor. Okay, so sensor role comes there only when you don't have that many resources, and sensors helps to sensors fulfill all these criteria. Okay, now this bioanalytical chemistry which help us to figure it out or develop the sensor, it is a sub branch of analytical chemistry. The other branches can be material, chemical analysis, or environmental analysis, about which you might be quite familiar. We will be discussing more about bioanalytical chemistry because it will be helpful for us to develop, understand the concept and terms used in the biosensors. Okay. Now, before we move to the sensors and all those things, we should have preliminary idea about why we need sensors. Now, before sensors came into existence, all these analytes or maybe the toxic component present in the environment or maybe in the healthcare sector, all these were routinely analyzed by different analytical instrument or devices. Here in this figure, I have just kept some schematic of some of those instruments which are routinely used to analyze either metal samples or maybe some volatile compounds or some non-volatile compound which are present in the sample. Now these technique, I'll just name them. Many of you might be knowing. The upper one on the right hand side is a high performance liquid chromatography. It's a separation technique as well as it detects the non-volatile component present in the sample. On the right hand side, in the same slide below, you have a gas chromatography, which is again a separation technique. It separates the different component present in the sample as well as it analyzes volatile organic compound quite efficiently at a very low concentration. Then on the lower hand side here, on the left hand side lower, you have this ICP-MS, which is the inductively coupled plasma mass spectrophotometry. Now this particular instrument is routinely used to detect the metal samples present in the different samples. It can be biological sample, it can be sample from the environmental sample, maybe from the military field and defense samples. All those kind of sample can be analyzed. Now, if you see the schematic and all this slide, you will come to know that all these techniques which I have shown here, which were routinely used for analysis over the years, although they are good in measuring the analytes, but they are quite complicated. They require large number of they need the solvent system, they need so many things. So it's not a simple thing. They are highly sophisticated instrument. They require a trained person. Also, they require high amount of sample preparation. You remember initially I told you about the matrix and all those things. So like if I wanted to analyze sugar from the blood, I need to see that nothing from the blood should be interfering in my sugar analysis. So that is known as the sample preparation. So sample preparation is a step prior to analysis, which helps to remove the interferences of all the matrix related issues. So all these techniques which are routinely used, they require high amount of sample preparation. To give you idea, like actually almost the 1/3 of the time and 2/3 of the cost which is involved for this analysis is utilized in the sample preparation itself. So you can figure it out how much these things are costly and not very affordable at our end. So that's why the need of sensor came into existence. Now, when these routinely analytical techniques were quite common, still we were facing some problem regarding analyzing the samples which were from healthcare or defense applications. So what was that common problem which we are facing? Like in a healthcare sector, if you see for sensor development, you need something continuous monitoring. So that was the first initial requirement in the healthcare sector. Then, as I told you, the analysis should require minimal sample preparation. sample preparation should not be a tedious or a laborious process which utilizes high amount of cost and all those things. Now, looking at our country and the kind of population we have large number of samples to be analyzed. Now, all these techniques which I have showed you earlier, they are relatively costly techniques, so we can't utilize them to analyze large number of samples. Now, again, the samples what we get to analyze, they are from the different origin, so they may have a different matrix, and again, there will be a different strategy to analyze them. Hello? 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 Hello?
Ma'am, you may please continue, ma'am.
Okay.
Sorry for
Yeah,
the
ma'am.
disturbance.
Sorry
No,
for
no,
the
no.
disturbance.
No. So basically in this slide, I wanted to give you idea that when we were trying to develop the sensors for the healthcare or defense application, what was that motivational factor behind developing the sensor? Because the routinely analytical techniques which were utilized for the analysis, they had some issues regarding continuous monitoring, or maybe they require sample preparation on a larger scale. And again, we have a limited resources. On top of that, there were strict guidelines by the regulatory bodies that this much amount of sugar only should be present in the milk, or maybe in the blood, or all those things, some strict guidelines from the healthcare sector. So to fulfill all those needs, it was difficult to manage with the conventional analytical techniques. And that's why we moved to the sensor development. Now, when we were discussing about the strict guidelines by the regulatory bodies, we need to understand at what are those regulatory bodies which governs or which decides that what should be the guideline if we are developing a device or a sensor for some healthcare sector. So these are some of the regulatory bodies which are looking after the development of a sensor in the healthcare sector. Like you all might be aware about All India Institute of Medical Sciences. Then the Central Drug Standard Control Organization is one of the organization which is dealing with regarding the monitoring regarding the sensor development in the healthcare sector. Then again, this Medical Devices Technically Advisory Group, which is basically a group from the Union Health Ministry. It also has developed some of the guidelines regarding the developing the devices for healthcare sector. And yes, this Drug and Cosmetic Act, which was basically in the 1940, again it was amended around something 2017, which discuss more about the medical devices. So these are some of the guidelines with which we need to be quite strict while developing the sensor. So to fulfill all these guidelines, we need to develop a device which can analyze large number of sample, that too with the minimal sample preparation and using our limited resources. So to do all these things, the solution was developing a sensor. So that's why sensors came into this complete picture, where they fulfill majority of the criteria which are required for the by that time of the requirement of that time. Okay, now you all might be knowing that since developing a sensor or all these things, they are quite interdisciplinary sciences. You need some input from the material sciences in terms of what material can be used for developing the sensor. A need of the chemical scientist or a chemist will be highly essential to develop the method or to do the trial and to do the, or maybe if you are developing some chemical compound which can sense your analyte, then biological sciences information will be highly required, because a biological component can sense all those compounds. Then the knowledge of physical sciences and all those things from the electronics and all those background is highly essential to properly make it a device. So it's an interdisciplinary science where the knowledge of all domain is highly essential. Okay, now you all are already might be quite aware, and already you might be knowing about sensors. Already you had 2 days session on sensor, but just in line with my talk, I thought let's give you quick little bit idea about the sensor. Now, normally when we talk about sensor, it can have different forms. It can be a basic lab-based sensing platform for detection. So whenever I say sensor, it need not has to be some probe or some device. It can be some method also. So like even if you develop a method to quantify some toxic compound present in the blood or maybe urine or something, that can be a sensor. So the sensor can be some lab-based platform for detection, or it can be as small as some wearable sensor where you are wearing it on a finger and then you can measure some of the metabolites or some parameters in your body. So the sensor has gone from the lab to the in the wearable form over the years. Now, in terms of advancement of the sensors, the advancement is limitless. You can see on the left hand side here, I have kept shown one small thermometer. Now, this thermometer is also not a primitive one. It gives you idea about the temperature in the digital form, that too up to the 3 significant figures. But this thermometer has evolved to these kind of a fever alarm armband. Now, this fever alarm armband, they were presented in 2015 IEEE this conference, and these are basically these pads which were attached to the body. They not only measure the temperature, but they parallelly measure the heart rate of the particular person also. Okay, so that much. Anyway, in my next few slides also, I'll be showing you different sensors developed across the years. But this kind of advancement has been occurred over the years in sensors. Okay, now I thought instead of keep repeatedly talking so much, let's quickly have some kind of a reflection point in between the slide. So this particular slide I have included. Now we are discussing about the high quality data. Like our sensor should be able to produce high quality data. That's what we always say. Now, what exactly does it mean when we say that high quality data? That data should be highly accurate and the data should be highly precise. Okay, so normally accuracy you can quantify by calculating error. The error can be absolute error, it can be relative error, and while calculating the error, what we do, we take the difference between the observed value, what we get, and the true value, which is the actual value of that sample. So by calculating that, we report the absolute error. Now, remember, whatever I am discussing in this slide, these are the very important parameters of the sensor. We are discussing right now that to generate high quality data, how we need accuracy and precision. I told you that for accuracy can be quantified by calculating error. Now, can you tell me how we can calculate precision? Is there anyone, or am I supposed to interact with you all, or maybe while I think they have kept a separate session for me for interaction. So maybe I'll leave this to you, that how precision can be calculated. Okay, maybe in our interactive session we can discuss about that. So basically, when we are discussing about the high quality data, we need to see that the data should be accurate and precise. Precision is basically deals with that how much repeatedly you are getting the same data. Okay, so you can just think of this thing, and maybe in our last session we can discuss that if we have to show that my particular sensor is giving me repeated results, what should be that statistical tool which I can use. Like for accuracy, I am using error. For precision, what it might be. Okay. Now with this, I'll move because I'll not interact you in between. Better, I'll keep that for that separate session which is kept there. Now from this slide onwards, I'll be discussing more about the sensor. Okay. Now this slide gives you idea about the sensor. So you have a sample. Now a particular sensor will have 3 components. You have a receptor, then you have a transducer, which will convert this reaction into some electronic format, then you have a signal processing unit, and then you have a digital display. So what actually occurs when a sensor come in contact with the sample? Now this blue one, what you can see here on the left hand side is your sample, and this sample contain n number of so many things. Now you can see this is my desired molecule. Can you tell me what might be the other name of desired molecule? This is my analyte, what I discussed initially. So I wanted to analyze this analyte from the sample. Now this analyte will react with my receptor. Now even if there are n number of things present in the sample, my receptor will have a high affinity to react with the analyte only. So analyte will react with the receptor, and this biochemical reaction will be converted into some electronic output with the help of the transducer, and then this will be shown in the form of a display. So that's the basic schematic of the sensor. Now the sensors can be of different types. We can have physical sensors which measure distance, mass, temperature, and pressure, physical parameters. Then we can have chemical sensors where the chemical sensors measure some chemicals which are present in the sample, like maybe the presence of some pesticide in the sample, or maybe present of some mercury in the sample, lead in the sample, or maybe this Maggie issue was there, quite common few years back. So present of those toxic chemical present in the food, that can be a chemical sensor. And the third category with which I'll be discussing more is a biosensor, which measures the chemical substances present in the sample, but my recognition element is a biological element. I'll again go to this earlier slide. So sample can be anything. It can be a chemical compound, but this receptor has to be biological. What might be the different forms of biological receptor, that I will discuss with you later. But if you ask me what is the difference between the chemical sensor and a biosensor, in chemical sensor and biosensor, the sample can be same, but receptor in case of chemical sensor will be some chemical compound, whereas receptor in case of a biosensor will be a biological component. Now all these sensors, they will be connected to a transducer, and then you get a response. Okay. Now when we are discussing about developing a sensor, we need to know what are the significant features of the sensor. Any sensor, even if you develop a sensor and you wanted to commercialize the sensor, you need to show that your sensor fulfills all these features. What are they? Accuracy and precision, I have already discussed. Accuracy is how much correct your sensor is able to determine. Precision is that if I'm using a sensor, how much repeated value I can get, or how much repeated value I am getting. Reproducibility is that reproducibility is a little bit broader term than the precision. It discusses that over the period of month or maybe over the year, how much reproducible values I am getting. Then specificity is that value which gives you idea that if there are n number of components present in my sample, my sensor should be able to only detect the analyte of interest. Even if I have not done any sample preparation, it should be highly specific. Now, to develop a specific sensor is very, very crucial. We normally have a selective sensors. That's why the next term selectivity. So maybe the sensor which we have developed is giving response to two analytes, analyte A and analyte B, might be more it is measuring more of the A less than that of the B, then we can say that it is a selective sensor. Now the next term in this is the sensitivity. Sensitivity deals with that how much low concentration I am able to determine. So that is the sensitivity term. Then the sensor developed should have a high throughput capability. That means that the sensor should be able to analyze large number of sample within a short span of time. And the most important thing, that the sensor should be relatively stable. Okay, so these are some of the significant features of the sensor. I'll repeat once again: accuracy, precision, reproducibility, specificity, selectivity, sensitivity, and stability. Again, high throughput. Now, one of the major feature of a sensor is a selectivity coefficient. I told you that it is very difficult to develop a sensor which is highly specific. So sensor will tend to give response to two analyte maybe. Like if you take an example, the glass electrode, which is very common to measure the pH. That pH combined electrode is commonly known as. So this combined electrode, which is used to measure the pH, it not only give response to pH or H plus ion concentration in the water, it also responds to the Na plus ion. So you wanted to determine H plus ion concentration, but your sensor is also responding to Na plus ion concentration. Now, how to deal with this? In this case, you need to know what is the selectivity coefficient. Or when you develop a sensor, you need to see that what is the selectivity coefficient value of my sensor. Now, what is the selectivity coefficient? Like when a sensor is used to measure ion A, but it is responding to X also. So the selectivity coefficient will give you relative response of the sensor to two different species. So on the numerator, you will have that interfering ion, whereas on the denominator, you have that ion which you wanted to measure. Now, the smaller the selectivity coefficient, less will be the interference. I hope you understood that how selectivity coefficient is important in developing a sensor. Now, you wanted to measure A, that's why the K value should be as less as possible. Now, there can be three probabilities of your K value. If your K value is more than 1, that it indicates that your sensor, here I have written ISE, I have written this ISE means ion selective electrode. Okay, so if my selectivity coefficient value is greater than 1, it indicates that my ion selective electrode or my sensor is responding to interfering ion than the analyte of interest or than the target ion. Okay, now if the K value is equal to 1, then I can say that the sensor is responding equally to both the ions. But if the K value is less than 1, then you can confirmly say that my sensor is responding excellent or selectively to the ion of interest. So to develop a good sensor, what you should have? You should have K value as less as possible. So selectivity coefficient plays significant role in developing a sensor. Now, can you determine selectivity coefficient experimentally? Yes, you can determine. If you see this particular graph, you can see that for mercury, you have a straight line. I'll be discussing this kind of graph later on. Whereas for lead and cadmium, over a large concentration range, you are not getting any response. This figure indicates that the selectivity coefficient for mercury is much higher than lead and cadmium. Now for the next few slides, I'll be discussing about chemical sensor. Let me figure it out how much time I have. Can anyone tell me how much time I have? Neha?
You have 10 minutes, madam.
Okay, for this session, I have 10 minutes now. Okay, so I have given you idea about the sensor and the basic idea behind the selectivity coefficient for a sensor. Now we will come to the first category, which is a chemical sensor. So these chemical sensors are miniaturized devices which can give you some real-time online information. And what we do here that as a chemist, we develop some material or we develop some molecule which can react with the analyte and that can give you a sensor. You all might be quite familiar with the pH strip or maybe some sensor which you just dip in the solution, you get different colors. So those are optical chemical sensors. Here I have just put a figure where you have a sensor kit which is commercially available to figure it out whether the particular water sample is clean or is it contaminated with the lead or is it contaminated with the mercury. So when this particular sensor solution or maybe a paper containing the sensor, if it is dipped in this particular sample. So depending upon which particular lead is present or mercury, it gives you color. And with this kind of a reference sheet, you can figure it out that which compound is present. So these kind of sensors are quite popular to measure this thing. So this is just a simple example. Now, when we talk about chemical sensor, the most common chemical sensor which comes in our mind is the ion selective electrode, because this is the most primitive sensor which is used. Now, this is a very basic example. This is a combined glass electrode used to measure the pH. Now, you all might be aware pH is the scale on which we can measure the hydrogen ion concentration, whether the particular compound is acidic or basic. You can figure it out using this electrode. So in our day-to-day life also, we have like with some of the acidic samples, some of the you have basic samples, so that you can figure it out. And this is a particular sensor. Now this sensor is basically a combined electrode where you have a working electrode and then you have a reference electrode. Now this working electrode is dipped in the dilute HCl, and the lower membrane of this electrode is, of this particular glass bulb is the sensing membrane, and this membrane is useful for exchange of the H+ ion across the membrane, and that is useful to measure the pH. So that's something about the ion selective electrode. Now this ion selective electrode, which is used to measure the whether the particular compound is acidic or basic, that also has some issues or some problem. As I told you, this particular sensor works excellent only within the range of certain pH. Like this is a normal pH range, broad range, but this sensor will give you excellent result only from pH 1 to pH 9. So even if you are going above pH 9, the sensor works somewhat abruptly and it gives you error value, and those errors are known as either alkaline error or acidic error. Now this why I have kept this particular figure, because I wanted to tell you that there are large number of problem which we may encounter when we are developing a sensor. So we need to take care of all those errors which are occurring during the sensor development. Now this is one example where I have developed this optical chemical sensor in my lab. So what we have done, we have taken a silica nanoparticle on which we have immobilized this potassium thiocyanate, and this is a reaction on which this sensor is developed. So this potassium thiocyanate, when it comes in contact with the ion, basically this is a sensor to determine the ion content present in the sample. That too, that ion should be in the Fe3 format. Now you might be knowing every element have their different oxidation state. So ion when present in the +3 oxidation state is known as a ferric ion. And this ferric ion, if present in any sample, when it comes in contact with this SCN, it gives you the bright red color. So what we have done, we have coated this thiocyanate on some silica nanoparticle, and this is the entire chemistry. So this is a basically nanoparticle based sensor. And when this came into contact with the water, you can see that for different concentration of the ion, you get different color, and just with your naked eyes, you can figure it out that what is the presence of ion present in the sample. I have also seen that even if this ion present in the sample, even if you have lead cadmium present in the sample, it is not causing any problem in the analysis. Now, what might be the application of this sensor for the healthcare sector? I have not done, but the particular sensor can be used to measure the serum ion test, because we are discussing more about the healthcare sector. Now, this test is routinely done where we try to figure it out that why iron content is less present in the particular human body, because iron deficiency may lead to large number of diseases. And we all know that a particular value of iron has to be there, maybe some 60 to 170 microgram for the proper functioning. So that's why these kind of sensor which I have shown you here can be extended application can be to measure the serum ion. Okay, so that is one excellent example of chemical sensor which can be used in the health sector. Now these chemical sensors or chemical biomarkers, what I was telling you, they can be useful to detect a large number of parameters, like you can measure the metabolites, you can measure the hormones or the proteins or the peptides. All these things can be measured where the sensing component is some chemical compound. So that's what the utility of the chemical sensor can be. Now, development of the sensor is not a single step. It contains large number of steps. So what are those steps that I have written here? You can figure it out that it is a very tedious process, and it involves the month of a years and so long to develop a sensor. These are some of the pictures which give you idea that how sensors can be in a liquid form, it can be on a paper-based device, it can be in this nanoparticle. So there can be different platform on which your sensor can be present. Okay. This is an excellent example that before your sample came into existence and after Hg come, how you can differentiate with the colors that how much of mercury is present in the sample. Okay. So in the last few slides, I gave you idea about the how the chemical sensors can be used for sensing. Now the next few slides, I will give you idea about the biosensor. I am thinking of skipping these slides right now because anyway, our next talk will be more about the biosensors. So there I will discuss more about biosensors, but try to understand that the difference between chemical sensor and biosensor is that you have some biological receptor here which will sense the chemical compound. And what might be those biological receptors? You can have enzymes, you can have antibodies, you can have nucleic acid. So these large number of biological components can be utilized to sense the chemical compound. The rest of the part of the sensor is same. Okay. Now, again, I have incorporated one question to just cross check, but I thought let it keep it for the interactive session. Now, this particular first session, I would like to conclude with this slide. You can see here, this is a very recent development. It's a pacifier biosensor. Again, it's a non-invasive saliva biomarker monitoring. You all might be aware that glucose monitoring is a very good biosensor where you need to prick with the needle your finger and that blood can be analyzed. So basically you measure the blood glucose content. Now, every time pricking and everything might not be possible, and that too for a diabetes, you might be knowing it's quite common nowadays with the very small kids also. So for those kids, every time pricking might not be possible. So these kind of non-invasive biomarker, these are the pacifiers. Now, this is a basic construction of the pacifier where this you can see the sensor is here, the sensing chamber is here, and this when the baby will put this particular first this kind of this portion will put into the figure. So with the saliva, it will move from here, and with this flow, when it will react here with the glucose oxidase enzyme which is present. Now this glucose oxidase enzyme will convert this glucose into gluconic acid, and that conversion can leads to the presence of H2O2, and that can be with the help of transducer, it can give you some signal in the mobile. So this much advancement you can see here, whether without pricking also blood glucose can be monitored. So this kind of advancement has occurred over the years in the healthcare sector, and this is very nice example where we can see that these devices can be used for very crucial issues like diabetes, which is quite common in the newborn babies also. So that's just the brief idea about the utility of the sensor. Now, if you see this particular graph, I don't know how much it is, you can see it because the figure is relatively small. You can see that with the change in the glucose concentration, the signal changes, and then thereby you can figure it out that how much glucose is present in that particular small baby. Okay. So that's one of the excellent example I thought of sharing with you at the end of this session. And that's what I would like to, I wanted to give you idea about this first section. Okay, thanks a lot for this session.
Okay, thank you, madam.
Okay, so what I am supposed to do, should I be there only, and then maybe because again joining might be again issue, so I'll be here only. And at what time we are starting the next session?
A 10 minutes break, madam. Be
Okay.
connected, don't leave.
10 minutes break, na? Okay, I'll
10
join
minutes.
then after 10 minutes, huh?
Okay, don't disconnect, madam. Be in the
Yeah,
call.
I am not disconnecting.
Okay.
I'm just unmuting my mic only. Okay?
Okay.
Let this slide be here only. Hmm?
Okay. Okay.
Okay.
Nisha?
Participants, now short break for 10 minutes. So don't log out your account. Please stay here. After 10 minutes, we shortly connect you. Thank you.
Hello? Hello?
Yeah, ma'am.
Yes,
Shall
Should
we
we,
start the session?
should we start, or you will tell me when to start?
Yeah, ma'am. Shall we start, ma'am?
we will start, madam.
Okay. Thank you. So, thank you all for attending this thing. And like as I told you in my first part of this talk, I gave you idea about the chemical sensors and biosensors. Now, you remember I told you that biosensors, the difference between the chemical sensor and biosensor is that the recognition component in biosensor will be some biological. It can be enzyme, it can be antibody, or it can be as a whole cell also. But that's not only the difference between chemical and biosensor. Now, biosensor have much more advantage over chemical sensor. Why? Because it's that tool which not only gives you idea about the how much compound is present, but it also gives you idea about its toxicological impact. I'll explain this thing to you. Like if you are using any sensor for that matter, what does the information that sensor gives us? That sensor gives us information that how much a particular compound is present in that sample. Okay, so maybe some gram or some concentration, some different units which we might be using to express the concentration, maybe some ppm or maybe milligram per gram. All those things we can figure it out. But what is the impact of that particular analyte on the living body, or to be precise you can say toxicological impact, that the sensor is not able to give us information. Now, when we are discussing about all these toxic compounds present in the environment and their impact on our health, and developing the sensor for healthcare, then we need to have those sensors which not only should be able to give us the idea about how much a component is present, it should be also able to give us idea that how much ill effect or the damage it is going to cause to our body. So that is the basic purpose, or that's where the biosensor differs from the normal sensor, that they give you idea about the toxicological impact of that particular analyte. Now, how they give you this idea, that I will tell you later on. Now, to understand what are the biosensors, this is the schematic which gives you idea about the biosensor. You have different compound present in your sample. Like in this sample, you have Hg, means mercury, or MP is a short form of a pesticide, which is a methyl parathione. So even if this red one indicates a pesticide, this is some other toxic component. Basically, these are two toxic compounds present in the sample. Now you have a bioreceptor here. As I told you, the example of bioreceptor can be enzyme antibodies. And with my today's presentation, I'll be discussing more about enzyme as a bioreceptor. So this enzyme will react with this analyte. It will only react with MP. Okay. That's why the choosing of the bioreceptor is a very crucial issue. So this receptor will only react with MP. Then this biochemical reaction through transducer will be shown here. And I have written here was this nanogram per liter. Can you see that? So this much low concentration you can detect using the biosensor. Now, what are the main components of biosensor? You have a bioreceptor and you have a transducer. On the different transducer, what we can have is a absorption, fluorescence, potentiometric, amperometric, about which you all are quite familiar. I won't be discussing much about the transducer. Rather, I'll restrict my talk to the bioreceptor. So you can have antibody, enzyme, nucleic acid, cell, molecular impregnated polymers as the bioreceptor. And I'll be discussing more about enzyme-based biosensor because they are the most primitive biosensors which are developed. Until today, they are extensively used. Enzyme as a biocomponent has a huge advantages over other biocomponent, as its availability, its cost is used, and that's why enzyme biosensors are quite popular among the different types of biosensor. Now, what are enzyme? Because many of you might not be from the biological background, so I'll just give you idea. Enzymes are the large protein molecule. that act as a catalyst. So you all might be aware that enzyme acts as a catalyst, and these enzymes can be used as a bioreceptor because of their specific binding capabilities. So enzymes act as a catalyst because enzyme has a affinity toward particular analyte, it acts with that. So because of this specific binding capabilities of enzyme can be utilized as a bioreceptor. Okay. Now, why enzymes are preferred over other biological receptors? As I told you, antibodies or maybe MIP, molecular impregnate polymer, because they have high specificity and selectivity. Now, what does this word means to us? High specificity means even if in your sample there are large number of compound, your enzyme will react to the only analyte of interest. That is known as a high specificity. And what is the sensitivity? Sensitivity is something to do with the concentration with up to which your sensor can measure. So normally majority of the analytes which we measure using enzyme-based biosensors, they are highly toxic compound, and that's why even if they are present at a very low concentration, their measurement is very important. So sensitivity comes there as a very important parameter. So if you say a particular technique is sensitive, it means that it can analyze very small concentration. So enzyme-based biosensor have high specificity and they can analyze small concentration of toxic compound. So whatever exercise I have put, I'll just skip through it because we wanted to utilize the time for the discussion. Or maybe we can discuss them later on. Now, this is a schematic which gives you idea about enzyme-based biosensor. Now, you all are aware, in a normal reaction, what we write? A plus B gives us C, okay? So, your A and B, when react, it gives you C. Similarly, in this enzyme, your enzyme is A, and your substrate is B, okay? So when your enzyme reacts with a substrate, it gives you the product. So this is P. So this is the basic principle which we are going to utilize to develop the sensor as an enzyme-based biosensor. Now this enzyme-based biosensor can be of two types. One is enzyme activation-based biosensor. What is that enzyme activation? Means in the presence of analyte, enzyme will be activated and it will give you more product. Okay. So this enzyme, this is your analyte. In contact with this, it will give you more product, and this you can correlate. This you can correlate that how much compound is present. So this is the figure what I have indicated here is a famous lock and key mechanism with which normally we express the enzyme activity. Now, why this is shown? Can you see this V shape? This is a very crucial part of the enzyme. Now, this is known as an active site, and at this active site, normally all reaction occur because basically these active sites contain some functional group which are responsible for the reaction. Now, here I have given you example of a urea biosensor. So when urea comes in the contact with the urease, this urease is an enzyme. It forms ammonia, and this ammonia can be detected with the help of the color. So if you are using urease enzyme in the presence of urea, you can determine the concentration of urea. Whatever the ammonia will be present here, that can be correlated with the amount of urea present. Now, here there is one more thing. You get a colored compound, and this colored compound is known as a can be used to determine the urea content. This is one example where enzyme activation based biosensor. These are not that much popular biosensor. The second category of enzyme based biosensor, which are known as enzyme inhibition based biosensor, they are very popular. Why they are popular? Because they in this what we do, we measure the activity of enzyme normally the way I told you initially. So enzyme in contact with the substrate, it gives you the product. So this is the first reaction what we do. Then what we do, then some toxic compound that will come into the picture. So this is shown here by blue color. So this is an inhibitor. What is the role of an inhibitor? It inhibits the activity of the enzyme. So this inhibitor will have somewhat similar structure like that of the substrate. So what it will do, it will fool the enzyme and it will react with the enzyme. Enzyme will think that it is acting with the substrate, but actually it will react with the inhibitor. Now when inhibitor will react with the enzyme, the active site will be blocked and the substrate will not be able to react with the enzyme. Okay. So what is done in the enzyme inhibition based biosensor? We measure the activity of enzyme before inhibition. This process, what I told you, is a inhibition. Okay. So we measure the activity of enzyme before inhibition. Then we perform inhibition process. And then we again measure the activity of enzyme. So whatever is the difference between the activity of enzyme before and after inhibition, that can be correlated with the amount of inhibitor. Now, why we are doing all these things, inhibitor before, after? Because majority of the inhibitor, say like organophosphate pesticides, they are not only pesticide, they are used as a chemical warfare agent. Even in the World War II also, they have been used extensively as a warfare agent because they have a huge impact effect on our nervous system. So that much importance these pesticides have. So in this particular figure, whatever we have shown you inhibitor is an analyte. And what is that inhibitor? It is an organophosphate pesticide. Why organophosphate pesticides are important? Because they are used in the World War II or in the World War as a because they block our nervous system, and that's why their importance or then sensing is highly important. And how we can determine this organophosphate pesticide? Using biosensor, we need to have enzyme inhibition based biosensor. So we will select an enzyme which can react with this inhibitor. Before inhibitor comes into picture, initially we react enzyme with the substrate. Now remember, every enzyme has its own substrate with which it reacts and it gives you a good product. So initially we'll react enzyme with the substrate. It gives us the good product. Then this enzyme will react with the inhibitor. So some sites of the enzyme will be blocked. Now this remaining site of the enzyme will again react with the substrate, and then you get a less product. So that thereby you can correlate the inhibitor concentration. I hope this is clear to you because this is the basic behind the enzyme inhibition biosensor. So you need to understand this. You just need to remember one thing, that there are certain compounds which are similar to substrate which blocks the active site. So, and to identify them or to quantify them, what we do? We measure the activity of enzyme without them, with them, and difference we try to correlate as their concentration. Now, as I told you, enzymes are highly important because they have an active site. Now, this is a diagram which gives you a schematic basically of an enzyme acetylcholinesterase. Now, this is a mammalian acetylcholinesterase. So, this is a biocomponent which we'll be utilizing in our biosensor. And this enzyme has some functional group present in the active site. Can you see? This is a deep well, and in which you have large number of functional groups which are going to react with the inhibitor. Now, in this figure, lower figure, I have shown you a simple reaction which gives you idea about what kind of reaction occurs. So, in the active site, you have this functional group of this enzyme. Now, this is the acetylcholinesterase, mammalian ACHE enzyme, and it is present in the brain. Now, when this enzyme come in contact with the organophosphate, it gets inhibited. Okay. Now, what is the first reaction? Enzyme with the substrate. So this is your substrate, ACHCl. Enzyme with the substrate gives you product. So you get some photon. Here in the step 1, you may get a huge amount of photon. But in the second step, what you are doing, you are not reacting the enzyme with the substrate. You are reacting enzyme with the inhibitor. So some of the enzyme site will be blocked, and here you get a reduced photon. You understand what we are doing? We are trying to find out that what was the initial photon count, what is the final photon count, and difference between the two photon counts can be utilized to identify how much of organophosphate pesticide is present. And why organophosphate pesticide is important? Because it is a chemical warfare agent. Now, when you are developing a sensor for this organophosphate pesticide, it is not a single step reaction. Rather, it contains optimizing large number of parameters. The different parameters which we need to optimize are pH and ionic strength, because you might be knowing enzymes are highly sensitive. Even if there is slight change in the pH or maybe temperature or maybe the salt concentration of the sample, the enzyme may loses its activity. That's why optimization of pH, optimization of temperature, even optimization of how much enzyme should be present, or optimization of how much substrate should be reacted, everything has to be done. So whatever I have shown you here on this slide is the work of almost like a year or more than that, where a person or a researcher who is working on this needs to optimize all these parameters for the enzymatic assay. So what you'll do, once you figure it out that which particular analyte you wanted to measure, you need to select from the literature that which is the enzyme which it can react with, and then you need to study the reaction. Like in this case, you have seen this reaction. Na? So this reaction I have taken as an example, and I optimize all these parameters for the reaction. So with those optimized parameter, now I am going to proceed further for the sensor development. Now, normally the enzymes, when they are present in the solution, they are highly disoriented, and that's why they don't give you reproducible results. And that's why what we do enzyme immobilization. What is enzyme immobilization? That I will tell you in the next slide. Now, when you perform these kind of experiments, the kind of plot what you get is a enzyme inhibition curve. And what is that? On the x-axis, you have an inhibitor concentration. Normally, inhibitor concentration is in the logarithmic scale, because you get this kind of plot we take in the logarithmic scale. And then you have here on the y-axis, you have a residual activity of the enzyme. So what you see that initially when the concentration of this pesticide is on the initial stage, when the concentration is very less, the activity is high. Whereas as the concentration of the inhibitor increases, the activity of enzyme decreases and it reaches to a stage where there is very less activity. So this is a peculiar S-shaped curve known as an enzyme inhibition curve. And using this curve, you can determine the concentration of the inhibitor. Now, the other term which I have written here, that LEDSD, you don't remember. These are some statistical term used to express the graph. Okay. Now, even if I told you that enzyme is a very good promising candidate to develop the biosensor, there are still some problem for using enzyme for biosensing application. The first one is the stability of the enzyme, and second one is the reproducibility of the results. So since I was facing all these issues, I thought that enzyme immobilization or coating the enzyme on some solid support can be one option, because in that particular stage, your enzyme will be highly oriented and then they will give you reproducible results. So there are different options by which you can immobilize the enzyme. You can just do physical adsorption of the enzyme. Here, can you see these are the 4 different ways by which you can immobilize. What is basically immobilization? It is coating the enzyme on some solid support so that you get a reproducible result. So the top one is just by the physical adsorption. This one is where you do some surface chemistry, some chemistry you do, and there is a bond formation between the surface and the enzyme. So maybe by chemical bonding. Then you have something known as a entrapment. And then you have something known as a, basically all these are entrapment example only. So these are the different ways by which you can keep the enzyme in a particular orientation so that you get reproducible results. Okay. So I faced few challenges also in immobilization, but with proper immobilization technique and all those things, you can confine the enzyme in a particular orientation so that you get a reproducible data. Now for the next few slides, I'm going to share some of my experimental observation. And with that, I'm going to explain you that how biosensors can be developed in the lab. Now this particular slide gives you idea about where I have immobilized enzyme on the zirconia nanoparticle. So I took zirconia nanoparticle on which I immobilized this enzyme ACHE. I already told you this is a acetylcholinesterase enzyme which is present in the brain and which is highly reactive towards organophosphate pesticide. Methyl parathine is an example of organophosphate pesticide. So when this enzyme comes in contact with the pesticide, it gives you some signal. So this way I performed the experiment and I could find out that if due to the presence of enzyme, how the activity of acetylcholinesterase changes. Now I use some colorimetric reagent or some reagent which helps me to identify the change in the reaction with the change in the color. So here you can see that with the increase in concentration of the pesticide, I'm getting different colors. So with naked eye also you can find out that what is that change in concentration or what is the different concentration what I have of the pesticide in the water sample. Now I measured or I use this particular biosensor to measure the methyl parathine present in the drinking water, agricultural runoff and seawater. Now why I took different sample? Because if you see all these water sample, they have a different composition. Like drinking water will be particularly free from any of the other interfering ions, where agricultural runoff may contain some of the fertilizers and all those things, and seawater will contain high amount of NaCl. So I could see that with this biosensor, even if there are n number of things present in the sample, my sensor is giving me good results and I can find out even 1 nanogram per ml of pesticide present in the sample. Okay, so this is a one example where you can coat or you can load the enzyme on the nanoparticle and you can determine the concentration of pesticide. Okay. Now this is another example. Now whatever I'm sharing with you right now, they are from my published work only. Now here I have used a different enzyme, a similar kind of enzyme, which is a butyryl cholinesterase. The earlier one was acetylcholinesterase. Now remember, whatever enzyme I have used, they are commercially available in the dry format. I have not extracted or purified from the living species. Normally enzymes are also present the way we buy chemicals. We can buy enzyme in the commercial live dry format. So those I have utilized for this sensor development. Why? Because they are highly pure and you get a good results. Okay. Now here in this slide, I have used butyryl cholinesterase enzyme. This is the active site where actually the reaction occurs. And why I move from acetylcholinesterase to butyl cholinesterase? Because butyl cholinesterase is relatively stable enzyme. My aim was to develop a sensor which should be able to give me consistent result over a long period of time. I also tried, this basically is a work from one of my co-author. I am also co-author in this paper, but this work was more regarding how you can stabilize the enzyme because enzymes are highly sensitive. So this stabilization of enzyme has been done in the dextrose sugar. And you can see that this particular graph, if you see here, on the y-axis, you have a residual activity. And on the x-axis, you have a time. So you can see that almost up to 6 months, the sensor was giving good results, which was relatively something good. Because normally sensors, the significant limitation of using sensor, if you say, is that they have a very short shelf life. Majority of the sensors you can't use for a longer time. But with this kind of a stabilization strategy, I can say that the sensors has not lost like from 100 to only 80, only 20% of activity has been lost. So still the sensor was good to use. Now this sensor was utilized to determine 3 pesticides. So you can see that this is a multi-analyte measuring device. So methyl peroxone, methyl parathione, and malathione. These are 3 toxic pesticides They are organophosphated pesticides, which are quite commonly used in India in the agriculture sector. And with this sensor or with this developed method, I could determine the concentration of these three pesticides in the same water sample, even in the milk also, in the concentration range of 0.5 microgram per liter. Now, why I wanted to explain you this thing, because from this regulatory bodies, there are guidelines that a particular compound should not be present above this limit. So there are certain guidelines, and this sensor satisfies all those criteria. That's why this sensor development was a very important thing. Now, whatever sensor was developed, the trend nowadays is to miniaturize the device, like from micro to nano and nano to pico. So that is our normal tendency to move. So initially I developed my method in the 96 well format. So this was my reaction vessel, you can see, in which small 96 wells were there, and I used to perform the reaction in each of these wells. Now, if you ask me, the capacity of each well was something around 200 microliter. So my entire reactant A, reactant B, and everything, it total accounts for 100 microliter. But that too was also a high thing, because enzymes are costly. Then you are dealing with the toxic compounds, so you should use them as less as possible. That's why I thought of miniaturizing the system. So I moved from this 96 well plate to 384 well plate and to 1536 well plate. Let me tell you, all these well plates are commercially available, and they are used to miniaturize your method. Like in this particular format, I used to use 200 microliter of the sample, entire solution. Here I can use only 20 microliter, and with this format you can use only 5 microliter of the sample, or maybe less than that. Now, one more thing, here you can use manually, you can work on this system, but when you have system like this, you need to have a automation. Okay, it's very difficult to manage all the parameters when you have a very small system. Now, I'm just sharing you my results. So this is one example where I developed a sensor on the chip. So titania chip was taken, on which I immobilized the enzyme by doing some surface modification. You can see here, and when this enzyme came into contact with the mercury in the sample, it gives you the difference in the response, and this difference in the response can be correlated with that compound. Now, in this particular example, we try to do real-time monitoring, whereby the detector what we have used was a charge coupled device, a CCD camera on top of the chip, which was continuously measuring the response. So whatever the change was occurring, we were able to figure it out. This was one of the example. This is not my work from one of my group member, where he has used the same enzyme what I told you, acetylcholine esterase. So this is a titanium chip on which there are different wells were done, and on each well he coated this acetylcholine esterase enzyme, and thereby he can find out that whatever the organophosphate pesticide is present. So these kind of enzyme-based sensors can be developed on the chip format also. Now, whatever I told you was everything on a batch mode. So it was not a continuous type of a sensor where the continuously your sample is coming and going. So the next example, which I'm going to explain you, is an example of a continuous flow-based biosensor. Now, when you have a flow-based biosensor, you need to have a system where the solution is continuously passing. So you need to have a pump and all those systems. And assume that this is your probe or a sensor. Now, the example what I have shown you here is a thermistor. What is a thermistor? Normally, as I told you, here I am utilizing enzymatic reaction as a tool to determine the concentration of the analyte. Now, majority of the enzymatic reaction are either exothermic or endothermic. They are either exothermic only normally. So there will be some amount of heat liberated with a particular reaction. Now, I need to have a database where I need to know that what are the different enzymatic reactions which can occur, and for each reaction, how much of heat is liberated. So with that data, whenever I perform a reaction for the unknown enzyme with the amount of heat liberated, I should be able to figure it out that what is the inhibitor concentration. So this is an example of a thermal biosensor where the heat of the reaction is utilized to sense the inhibitor concentration. Now, to tell you how the sensor can be developed in a continuous mode, you understand that this is the basically your device in which you have a probe, in which you have coated your enzyme on some, here I have coated on the control pour glass, which is a very inert material. You can coat the enzyme on that. Now, when the solution, your solution through the pump comes, and when it comes in contact with the enzyme, then here the biochemical reaction will take place and some heat will be liberated, which will be sensed here. Now, what will happen when there is no pesticide? The heat liberated will be more. When the pesticide will be there, the heat liberated will be less. So the difference in the heat liberated can be correlated with the amount of pesticide. Again, the same enzyme we have used here, acetylcholine ester, which is coated, and that gives you the result. Okay, so these kind of results you can get here. Even this particular sensor can be utilized to determine the choline. Now, choline is a very important component present in the milk, like and it is required for our all nervous system development. So this is an example of a choline sensor, which is a continuous type of a sensor, and you can determine choline content present in the sample from 0 to almost 1 millimole per liter. So that's the range within which your sensor develop, and these kind of peaks you can get when you have a different concentration of a choline. So normally what we do when we develop a sensor, and once you figure it out that under this condition your sensor is working absolutely normal, you take different concentration of your standard solution, and for each of them you get the signal, and then you plot a calibration plot where for the different concentration what response you are getting, and that plot is basically a guideline for you. So using that plot you can always identify that what is the concentration of an unknown. Like say if you take an unknown, with that unknown you will measure, get some signal, and then you will go to this graph where you have already data with the standard solution that for which concentration you are getting how much response, and thereby you can figure it out that what is the concentration of the unknown. Okay, so that is about my work which I have discussed you. So I discussed with you basically 2-3 enzymes and how you can immobilize them on a different platform. It can be in a batch mode, it can be in a continuous mode, and you can utilize them for a biosensing. Nowadays, the current trend is in the microfluidic devices development, where you have a substrate and a buffer different ways on the same platform, and then you have a mixing channels, and then the enzyme will come from here, inhibitor can come from here, and then you can see. So this is basically a schematic. You all might be aware about the microfluidic platform, so I'll not discuss more about that, but try to understand that from one reservoir your pesticide will come, one reservoir your enzyme will come, and when they will react, the detector will sense the activity of the enzyme in the presence or in the absence of pesticide. So that's the application you can definitely find out when you are discussing about the defense application of the sensor, where all these organophosphate pesticide are a chemical warfare agent. Okay, so now there are large number of difficulties which may occur while developing a sensor from a micro to nano scale, but for all these things, if you have an automated system, then you can definitely take care. So majority of the laboratories in the developed countries, you will see that they have an automated system where all the things are monitored by computer, and thereby the timing of the adding the solution and measuring everything is automatic. Okay, now the last few slides of my presentation, they are regarding the advances in the chemical or biosensors, and here I am going to share a few of the interesting example where the sensors are like wearable sensors or chip-based sensors which are developed over the years. Okay, so just look at this figure, and then I will discuss you that what I wanted to explain you here. Okay, so if you see this particular slide, you can see that on top you have a blood glucose monitoring device, which is quite common. Many of us might be using because diabetes is now a very serious issue. Now, this particular device, which is commercially available, it basically is an amperometric biosensor where the glucose oxidase enzyme is quoted here. Here, if you see the here where you keep your drop of blood, na, there you have a glucose oxidase enzyme. This enzyme, when comes in the contact with the blood glucose, it forms a gluconic acid and hydrogen peroxide. And this hydrogen peroxide forms H plus and O2. And this reaction used some readable format through transducer. So this is a sensor. Now, with the advancement, now we all are quite familiar with this kind of a finger pricking device, which is used for measuring the glucose in the healthcare sector. Now, what is the advancement of this? Every time pricking might not be possible. Even if it's the adult, then again, it is not very advisable. So you have, now there are certain patients for which the glucose level is abruptly changing over the period daytime. And for them, monitoring is highly important. So you have something known as a CGM, continuous monitoring glucose monitoring device. It is a needleless device. It's a kind of a patch which is connected with the mobile, and whatever is the change in the glucose concentration over a period of time or from time to time is sent to this mobile. And through this mobile, you can see that what is the change in the glucose concentration. Now, the slight advancement of this is also, this is when you have a tattoo sensors. I'm from Goa, and you all might be knowing that tattoos and all those things are quite common in Goa. But this tattoo technology is also utilized for the sensing purpose. How? Because this, if you see this MIT, they have developed a ink. Now, this ink contains some genetically modified living cells, which gives the response with a particular analyte. And this ink is used to form the tattoo in the form of a tree. Now, can you see a tattoo here? And in this tattoo, you can see 3 different colors are there. What does these 3 different colors gives you idea about? These 3 different colors is example of a multi-analyte tattoo sensor. So if you have 3 analyte in your sample, for each of those analyte, this ink will give you different color. And with the help of that color, you can identify that what is that compound present. So can you imagine how much advancement in the biosensors in the healthcare sector has been done? From pricking sensors to a patch on the body to the tattoo. Like even the ink can be of some biological component, and this ink, each branch of this tree, you can see that it is sensitive towards the different chemical compound or a molecular compound. That is the next generation wearable devices. Some large amount of work is working there. There are certain groups in the USA which are working on this. Now, one more example I wanted to show. This is not an example of a biosensor, but this is a example, you can say, as an example of a physical sensor. So nowadays we all are quite particular about diets, and you wanted to see, track how much meal we are taking, how much. So this kind of a wear scan is a belt which is developed by the UCLA scientists. And what is done here, when you wear this belt in your neck, so then this vibration data which is generated in near our neck, that is transmitted to the smartphone, and that gives you idea about how much you have taken meal and what are the contents of your meal. So all those things can be stored in the mobile, and that will give you idea about your health status. Okay, so some of the sensors which I thought is nice to share with you, I have just shared it with you. And this is the one more, like, I just wanted to show you that it's not only with respect to the healthcare sector or with the, then sensors can be used in the food technology also. Basically, ultimately, we are going to use this food only, and then finally, if the particular food contains toxic compound, then maybe getting affected by that. So these kind of sensors which are also developed, which can determine the whatever the toxic compound present in the body. Not only toxin, what are the important compound present in the sample, whether they are in an optimum amount or not. So all these things can be figured it out. So that's all about the development of the sensors over the period. Okay, so what I was discussing with you in the last few slides, I will again quickly go through this. From here, I told you that what are the different variable biosensors. Now, variable biosensor itself is a new branch of biosensor where majority of the sensors developed, you can wear them and then you can measure them. Now, these are the advancement what I was discussing. Even if nowadays many people wear the sensor, the watch, smartwatch, that also is an example of a variable biosensor. Okay. I just forgot to discuss some of the important thing regarding biosensor because I thought it will be too much for you to understand. But just one example, I would like to explain you that how this kind of enzymatic assay can be useful. So I'll share a very simple figure to give you idea that how these sensors can be utilized. So let's quickly see this particular slide. This was the example where I told you that acetylcholinesterase enzyme is utilized to measure the organophosphate present in the sample. Now what happened? This is that enzyme inhibition curve what I was telling you. Okay. What happened when the inhibitor concentration was less? The inhibition was less. Now on the y-axis, initially I told you, you can have different things. The figure I shown you initially, which is the S-shaped curve, at that time on the y-axis you have a residual activity. Now when you have a residual activity, your plot will be something like this from top to bottom. But if you have a percentage inhibition on the y-axis, your plot will move like this because residual activity and percentage inhibition are inversely proportional. So that is one thing I wanted to tell you, that based on what you have on the y-axis, your curve will either start from the less value and will go to high or from high to low. But always remember the shape of the curve will be that like that only, S-shaped curve only, because over a period of time, the response is going to change that way. So this particular figure, if you see, you can see that from concentration 0.01 to 100 nanogram per ml, I can determine the concentration. And now this particular sensor not only gives me idea about the amount of organophosphate present in the sample, it also gives me idea that how much of inhibition is occurring. Now I was telling you that this kind of biosensor not only gives idea about the concentration, but they give you idea about the toxicological data. What is the toxicological data? It is an inhibition only. What is the inhibition? When I take 100 cells initially, and when that 100 cell comes in contact with some toxic compound, few of the cell will die. Now that how many cells will die when they come in contact with the toxic compound is an inhibition. So that way, this entire study can occur. I'll again repeat you that you might not be, I don't know how many of you might be able to understand the inhibition concept. Basically, here we try to find out that what is the impact of the toxic compound on the living cell and how many living cells are live or dead, and that we try to correlate in terms of the percentage inhibition. So what is the inhibition? The cell which are live and the cell which are remaining live after the pesticide come in contact. So that is something I wanted to discuss with you, and that's why I again told you all these things. So we discussed two types of biosensor. Basically, when the activity of the enzyme increases, it's enzyme activity biosensor. And when you discuss with respect to the defense field and all those chemical warfare agent, you need to have a enzyme inhibition based biosensor. Okay, so that's what I wanted to discuss with you. These are a few of my references which I wanted to share with you, or maybe some of the references which I have used for preparing the slide. Now, at the end, I would like to tell you that why we need enzyme-based biosensor, because they are sensitive, they require minimal sample preparation. Then you can analyze large number of sample. You have seen those well plate format, 96 well plates. So, and I can analyze almost 96 sample at the same time. Now, if you remember, I told you I used to use some 100 microliter of solution only. So I use very small amount of enzyme. So cost wise also they were economical, and there has been advancement over the year where these type of biosensor can be used in the field also. If you are interested, I will share you that results also. And they give you good precision or good reproducibility and good accuracy. That's why the enzyme-based biosensor, even if they are the primitive one in developing this biosensor, they are still used as an excellent tool to determine large number of toxic compounds from the healthcare sector sample, for the clinical sample, as well as from the defense, all this. Okay. Now, these are my references which I have utilized to prepare this slide. Some of the references I have taken from other source, and that's what I wanted to share with you regarding my work in this next session. Okay. So that's what I would like, I was planning to share with you. Hope you enjoyed both the session and you understood what exactly this sensors, my journey through this sensor development, and how we develop different biosensors on different platform. The platform which I discussed was on a chip, on a nanoparticle, or maybe on some probe. So all those things you can definitely try out when you develop. Now, just because this is a FIP and all of you are faculties like me only, just one question came into my mind, and I thought that let's share this thing so that I can get some input from you all also, that how we can use the sensor in our education or classroom teaching. That can be one thought which we can think, so that this knowledge of sensing can be utilized for our educational purpose also. It should not only restrict to the research, rather than it can be utilized on an educational front also. So with that question, I would like to conclude this session. I hope you all enjoy this session.
Yes, ma'am. Certainly, your presentation is very excellent, ma'am.
Thank you, Priya. And I am really thankful that you gave me opportunity to, because of this, at least again, I could also get so many, I can be in touch with you all people, so that it's really nice to be in touch with you all. Thank you.
It's my pleasure, madam. Shall we continue, or
it's better if we can continue, so that everyone can be free. Maybe they also might be like throughout the day, they might have done teaching and all those things. So if you have any queries, you can ask me, or then otherwise I have some questions just to so that you can remember whatever I have told you. So anything is fine with me. If anyone has any question, you can please ask me. Please feel free to ask me any questions. I'll keep my presentation like this only, so that if you don't have any question, we can quickly go through the content, so that it will be better for you. Let me tell you what you prefer. Because, okay, let's start with this question. Can anyone give me idea that how we can incorporate sensors in the educational classroom teaching? Let's have a discussion only, because you all are so senior, and so I just don't want to ask questions, just a discussion kind of thing, so that at least I'll come to know so many good things from you all.
Nisha, please unmute the participant's mic. Rahul?
Good evening, ma'am.
Give access to the participants.
Yeah. Now we can able to access. Good evening, ma'am. This is Dr. Premkumar from Siddharth. Actually, the session is really useful, and I would like to start a few questions regarding chemical sensors.
Please.
You will be discuss about the basics of chemical sensors today very neatly. I would like to know, actually, we are working the projects like industrial chemical outlets.
Yes.
So what kind of sensors will be used to, detect the chemicals in outlets in industries?
Yeah, so basically,
can you say a few examples so that each and every participants can come to know, and we can have a possibility to work on that also?
Sure. Like, I'll just take the example from my samples only. Okay. So, few samples, like if I have a industrial effluent from the metal industry. Okay. So, which contains some metal ions, maybe lead, cadmium, and all those things. Now, I am sharing a very, very basic data here. So, first thing I need to know that what is the basic chemical composition of my industrial effluent. If I know that particular effluent contain, just take an example right now, that it contains large amount of metal ion where mercury is the significant contributor, and I wanted to develop a sensor for a mercury. Now, in that respect, I need to develop a ligand which will bind with a mercury slightly different than the other ligand. Now, can you see here on this slide on the left hand side, I have a ligand which is developed. So this work I have done with collaboration with an organic chemist, one of my colleagues. I am an analytical chemist basically. So my work is to develop a method. So we developed this ligand, and this ligand, then we reacted with the different solution which contain different metal ion. You can see here, this was a lead, then we have cadmium, then the calcium. So all the solutions we have taken and we added this ligand. So the color of ligand was different initially. And when it come in contact with this different metal ion, it gives you a different color. So with mercury, it was giving entirely different color. So that way we can figure it out that this sensor is selective towards the mercury. That is one approach. Then we thought of developing a paper-based sensor where we immobilize this ligand on the paper, and then maybe on the nanoparticles, where these nanoparticles can be utilized to give you the color. Like here I can show you, like here this potassium thiocyanate we have coated on the silica nanoparticle, and when this potassium thiocyanate comes in contact with the any solution containing iron, it gives you red color. So these are the solutions which contain high amount of iron, and these are the solution where you don't have any iron content at all. So basically the first thing will be to develop a ligand which will bind effectively with your analyte of interest. And once you are sure about that ligand reacting, then you need to take care of the other parameters which should not interfere in your analysis. That's the strategy how you can work. And when you are sure that within that particular framework, your ligand is effectively reacting with the analyte, then you can go forward and immobilize it on a nanoparticle or maybe on some other paper so that it can be used easily. So that is the strategy. I hope I could answer your question.
Yes, ma'am. Thank you so much. And one more question. Like your discussion, you are shown education how the sensor will be used. In our college, right, we are using for PAR sensors. So in the classroom, in our college, we are using for PAR sensor in the wall.
Okay,
so
okay.
Whenever the object, it detects all the electrical appliances, it will be automatically turn on. And if there is no persons which is available in the classroom, so automatically the appliances get turned off. So
great.
we
So
can
you
able
are
to
doing
use
energy,
our energy saving applications. We can able to use for like the PAR sensor and all.
Great. So I'll at least forward this note to our head that we don't have these PAR sensors here. So definitely with your this thing, we can definitely plan to have these sensors.
Yeah.
Anyone else would like to ask some questions?
And one more, this kind of a sensor design and all, what are the list of softwares you people are using in your laboratory?
To tell you frankly, I am a chemist, so don't use much software at all. We work in the lab more. So just to plot the graph and all those things, we need software. That's the only application of our sensors, what we use. So in that front, I won't be able to give you much data. Like for my data calculation and all those things, I use Origin or Excel only. Not more than that. So I am not an engineer person, not much tech in this software. I don't have much idea.
Okay, thank you, ma'am. Yeah.
Anyone else would like to ask any question? I hope already all of them are tired. So what I will do, I'll just quickly go through a few questions which I thought, so that at least I'll come to know that how much people have, at least, because it was in a very short this time frame. I discussed so many things. So let me quickly go through some of the questions which I thought. Is it okay?
Ma'am, I have one question.
Please.
In glucose monitoring device, the blood is the source. From that blood, you can measure the glucose, is it?
Yeah.
But in the tattoo type sensor, from what is the source for that sensor to measure the parameters?
Which sensor? Which for?
Tattoo sensor.
Type 2?
Tattoo.
Tattoo sensor, na? Tattoo, na? That ink which is used to make the tattoo, na? In that ink, some biological component is there.
Ink will react with what?
Yeah. So you assume that I'll just go to the slide. I'll show. Now this ink, na? This ink is not normal ink, okay? I just assume that this ink is a combination of a ink plus some enzyme solution, okay? So this ink contains some enzyme solution in that. Now, when this ink containing enzyme is coated on our skin, whenever this enzyme comes in contact with some toxic compound, it will give you color.
Toxic compound means sweat, from sweat it will identify.
Anything like assume that what enzyme I discussed with you, acetylcholinesterase enzyme. Now remember, just remember for this particular slide, acetylcholinesterase enzyme is that enzyme which, when reacts with the organophosphate pesticides, give you some signal. Now that signal can be anything. Here say color change, okay? Now assume that this particular enzyme, when reacts with the organophosphate, give you signal. Now in this ink, if I have added this enzyme, now when my skin comes in contact with the pesticide, what will happen? This will give me some signal. Now pesticides are normally colorless, you can't make out, but when my skin will come in contact with the pesticide, either the green or a yellow, red or a blue color will indicate that some amount of pesticide is present. Now here I have shown you a very advanced example where I can have three compound, like I can have a pesticide, I can have some mercury, I can have some other compound also, and for three of them also, I'll get three different signals from the same enzyme. This is a complex example I have taken here, but to explain you, I can tell you that the ink contain enzyme. The enzyme react with the with any whatever you wanted to measure, sense some of the compound from the healthcare, like maybe sugar also you can take. Sugar also, if it reacts with enzyme, react with the sugar, it gives you some response. And here the response is in the form of an optical response, so that color change can be identified as a response. Is that fine?
Okay. Okay. Thank you.
Okay. So I thought there are no questions. Maybe I'll forward you the questions, whatever I have. So with this, I would like to conclude.
So, is there any possible to take up any virtual industrial visit to your laboratory? Possibilities I'm asking. Yeah.
No, it's not at all about it. I am right now in a Goa University. I am right now in the biochemistry department where we don't have these facilities. I have done majority of my work in the BITS Pilani Goa campus in the biosensor lab. I will just check on the website whether they have any of the clip for that sort, and if it is there, I will share with you this thing. But whatever work I have shared with you, that I have done in the BITS Pilani Goa campus, there we have a lab. But right now, where I am working, there I don't have these facilities.
Thank you, ma'am.
Okay.
Participants, do you have any doubts? Please ask.
I think we
I
can
think
conclude
no
this
questions.
session.
Okay, madam. You proceed.
Okay, so thanks a lot. I initially I was thinking of asking, discussing some question, but I thought already it is 2.5 hours more than that. So better I will send you question for the exam, and thank you for giving me this opportunity. And if I get some link, what one participant asked me about the question virtual tour, I will definitely share that link with you. Thank you.
Thank you, madam. Thank you.
Okay. So can I leave this session now?
Okay, madam. We can do.
Okay.
Thanks for your valuable time with this.
Thank you, Priya. And thank you everyone for patiently listening and all those things. Okay. Thank you. These are my references. If you need, let me tell you, this is okay. Just to before leaving, this is our Goa University. We have so I am from there. So I thought let's share this slide with you. And that's what with that I would like to conclude. Okay. Thank you.
Nisha?
Yeah, ma'am.
Conclude the session.
Ma'am,
Is
next
Rahul is there? No interactive session.
Okay.
Completed.
Just shall we proceed the formal vote of thanks?
Okay, ma. Before that, we have to post that link now. Google form.
Yeah.
Is Rahul is there?
Wait, ma'am.
Wait. Just a minute.
Participants, please wait just a minute.
Anisha, I have posted the link.
Okay, ma'am. Participant, one small info. We posted the link about your attendance. Please fill the Google attendance. This is we are taking for your ATL FDP attendance. Please fill it. It is in the chat box. Please fill the Google link. This can be considered as your attendance for the ATAL FTP. No need to post your ID in the chat box. In the chat box, Dr. Priya ma'am posted the link. The Google link is for your attendance. This can be considered as your the 5-day FDP attendance for the ATAL offline session. Thank you.
Participants, is there any difficulty in filling the form? Participants, please respond. Anybody?
There's no difficulty, ma'am.
No, ma'am.
No, madam. No, madam.
No, madam.
Okay, sir. Okay.
I hope today's session is very interesting about the biosensors, mainly about the tattoos, that and all we can consider into a wearable sensors and all. It's very interesting. So I hope the same enthusiastic should be continued for the tomorrow session also. We will meet you on tomorrow sharply 6:45 p.m. Thank you everyone for your cooperation. Bye. Good night.
Thank you, madam.
Thank you, madam.
Should I close the session or wait until 9:30?
Doctor Kosubendra, shall we end the session? Stop recording.